This is lecture 5A, part 1. It's on the second topic in cardiovascular physiology, which is the vascular system. And in this photomicrograph, what you see is actually quite a informative photograph of tissue showing a capillary, and you can see that if you look carefully, you see that the capillary is looking, that's one branch of the capillary, that's this other branch of the capillary, and they're joining up here and flowing that way. And the thing to note in this is that capillaries essentially are about one red blood cell. They have a diameter not much bigger than a single red blood cell, which means that here's a red cell, and on average a red cell would have a diameter of about 8 microns. So you can see that they're quite tiny. They're all through all the tissues, and this is where you get exchange of gas, nutrients, and metabolic wastes between the tissue and the blood, and vice versa. All the rest of the vascular system has to do with transportation. There's no exchange going on anywhere except in capillaries. The idea is to carry the blood into close contact with every single cell in the body. There is one type of tissue that doesn't have its own blood circulation, but is close enough to the blood that it gets enough oxygen, or it's close enough to the surface that it gets enough oxygen, and that's all epithelial tissues. You have 4 major types of tissues in your body, and one of the 4 are epithelial tissues, and those tissues don't have their own circulatory supply. So, in general, the circulatory system involves all of these parts here. When it leaves the heart, the blood is in the arterial system, and then it gets into arteries and smaller arteries and arterioles, and eventually to a capillary bed where the exchange occurs. So exchange is occurring in this area, and then it goes into the venous side of the circulation and flows back to the heart. So a blood vessel going back to the heart is part of the venous return. It's a vein. A blood vessel going away from the heart is part of the arterial circuit, or part of the circulation. Overall, the generalized structure of blood vessels is different between the venous side and the arterial side. And here we're looking at sort of an average-sized artery and an average-sized vein. There are some differences when you get larger or smaller arteries and larger or smaller veins. But in general, you can see all of the main features here. There are 3 layers. One is the tunica interna, which is the innermost layer. It's sometimes also called the tunica intima, I-N-T-I-M-A, intima. So there's the tunica interna. The next layer going out is the tunica media. And the 3rd layer is the tunica externa. And if you look carefully, both the arterial side and the venous side and an average size blood vessels have all 3 layers, but there are differences between the arterial side and the venous side. In terms of the innermost layer, the tunica interna, there is a single layer thickness paving of the interior of the blood vessels. It is made of endothelial cells, and so together they all form an endothelial layer or an endothelium. This is all part of the tunica interna. So the walls of the surfaces of the blood vessels that's in contact with the blood itself is paved with these cells, endothelial cells. Just underneath that you have a layer of connective tissue, which is not made of cells but secreted material, and we talked about it before when we were talking about collagen just underneath, the cells that line the blood vessels, and that collagen is a connective type of material that holds things together, and we talked about it when we were covering coagulation. So that's the subendothelial layer, also sometimes called the basement membrane, or collagen, made of collagen fibers. Outside of that you have an internal elastic lamina. Okay, this is elastic type of tissue. You have that internal elastic lamina only on the arterial side, not on the venous side. This helps the vessel hold the higher pressure blood that's present on the arterial side of the circulation, helps keep the vessel intact. Just outside of that you have a tunica media, which is made of muscle tissue, smooth muscle tissue. And the difference between the arterial side and the venous side is that the smooth muscle is much thicker on the arterial side, again to be able to retain that high pressure blood that's flowing through. There is some smooth muscle and a tunica media on the venous side, but it's much thinner. Then outside of that you have an external elastic lamina, again only on the arterial side, not on the venous side. The walls of the veins, in fact, are fairly, soft. They bulge out easily when there's more blood present. Of course, the thing that makes the arterial side vessels bulge is the pressure wave that occurs every time the heart contracts during systole. And then outside of all that is the tunica externa, which is a connective tissue layer with nerves and blood vessels, things like that. Okay, so we'll go through that, essentially what I just said. The blood, of course, as I said, carries the blood away from the heart. Walls of the largest vessel on the arterial side is the aorta. It's dominated by lots of elastic fibers, allowing it to, pulse but retain that high pressure blood within it, but it can pulse. It has a lot of ability to stretch out under pressure and then recoil back. Lots of smooth muscle and elastic fibers, both an internal elastic lamina and a external elastic lamina. The thing about arterial arteries is you can actually see right away if it's an artery or not, because you can see the thickness of the wall. You'll see lots of smooth muscle. You see a thick smooth muscle layer, and that smooth muscle layer is shown here all the way around. All of that is smooth muscle tissue, all the way here and here. All that area there is smooth muscle. And the other thing you can see that easily identifies this as a artery. Let me just get rid of this. The other thing you see is you can easily see the internal elastic lamina, because it looks like a elastic band, like a scrunchie, that women will sometimes put in their hair, which is an elastic band itself. And you can see it here as this sort of wiggly line. That's the internal elastic lamina. So that's very, very obvious in this. That's a giveaway that you're looking. That's the other giveaway, the key typical characteristic of a reasonably sized artery. I'm not going to draw it all the way around, but that's the internal elastic lamina. This is a blood vessel that's been cut in cross section, a thin slice of tissue put onto a slide, and then a photograph taken. So it doesn't have any blood in it. There's nothing in it under high pressure. There's nothing there to stretch it out. But you can see that the internal elastic lamina and the external elastic lamina, which you can't easily see at all, but is probably somewhere in this area here, okay, just outside of the tunica media, the muscle there, both help handle that high pressure and allow the vessel to bulge and expand and then and recoil. So here you see again the in this case it's a drawing. The when you look at it overall, the arterial side of the circulation, these are resistance vessels. They have a certain resistance to flow because they have that characteristic of less easily distended blood vessels that have a thick elastic muscled wall. The elastic fibers allow the vessel to withstand high pressure, and the smooth muscle allows it to contract or relax. And contraction is called vasoconstriction, and dilation or relaxing is called vasodilation. So that allows for change that allows, because you have that muscle that can contract or relax, and by the way, all that muscle there is smooth muscle tissue. There are 3 types of muscle in the body: skeletal muscle, cardiac muscle, and smooth muscle. And it is, of course, under involuntary, unconscious control. You don't consciously control your smooth muscle tissue, and therefore it is controlled by the autonomic nervous system, which, as you well know, has 2 divisions, the sympathetic division and the parasympathetic divisions. So blood vessels can contract, vasoconstriction, and dilate, vasodilation. Mostly it's the arterial side, it's the arterial side of the circulation, particularly the arterioles, that are able to vasoconstrict and vasodilate. And so they can change the overall blood pressure. You can have changes in diameter of the blood vessels systemically everywhere, or it could be just restricted to one particular system in the body, or even one particular area of the body. So you have the body has the ability to change the amount of resistance to flow, and therefore can change the blood flow to a particular area if it's restricted to, if the change is occurring in one area. Or if the change occurs everywhere, then you can control overall blood pressure, because if you get vasoconstriction, then that gives less space for the same volume of blood, and so pressure rises in that case. If you have generalized vasodilation, pressure will drop. Here's a diagram of some of the major vessels on the arterial side of the circulation. There are many, many names here, but only a few of all the names in the body. And if I don't mention it, I wouldn't expect you to know the blood vessel. We have covered a few of them, however. We've talked about the brachial artery in the arm. We talked about the radial artery, which is in the wrist. We talked about the left and right common carotid arteries. We talked about the aorta, and it has an ascending portion and a arch portion, and then it dives down and becomes the abdominal aorta. Let's see if they've marked it that way. Yes, the abdominal aorta. Before that, just before it goes through the diaphragm, it's called the thoracic aorta, and then the abdominal aorta. So we've talked about all of these so far. Well, arterioles are the smallest arteries. Remember, on the arterial side of the circulation, getting pumped from the left ventricle out into the systemic circulation. There's two major circulations. I'll show that to you quite soon. There's the pulmonary circulation. There's the heart here, right? So there's the pulmonary circuit, and then there's the systemic circuit, which goes everywhere else in the body. So the pulmonary circuit goes up to the lungs. The systemic circuit goes everywhere, including the, all the other tissues, the head, the neck, everywhere. And the pulmonary circulation is what allows blood to go from the heart up to the lungs and then come back to the heart. So it comes into the left side, comes into the right side, gets pumped out of the right ventricle up to the lungs, and then returns back to the left atrium, goes to the left ventricle, and gets pumped out to the rest of the body. So the systemic circulation is much larger, under much higher pressure, and in that, in any part of the arterial circulation, you have arterioles, which are the smallest arteries, right? Getting back to the slide itself. And these arterioles are the main point in the circulation where you have most of the activity in terms of vasodilation and vasoconstriction occurring to change blood flow to different areas or to change the overall blood pressure in a regulatory manner. The capillaries themselves are the very thinnest vessels, and I showed you that initial photomicrograph on the first slide, which indicates just how thin and tiny they really are. The walls, in fact, are 1 cell thick. They are paved as are all the blood vessels in the body. They are paved with endothelial cells, but that's it. Underneath the endothelial cells, you don't have any other cells. You do have a connective tissue layer, the basement membrane, or what was the term, an endothelial, subendothelial layer, but there are no other cells. It's extremely thin, and this is the exchange. This is where internal respiration is occurring, exchange of gas and materials between the blood and the tissues. And here you see it again, the same photomicrograph. I should point out to you that you can also see very easily the endothelial cells. This is, I'll do it in another color. This is a nucleus of an endothelial cell, and the entire endothelial cell is roughly, I'm outlining it here, paving that portion of the wall of the capillary. And then there's another one, there's the nucleus of another one. Here on the other side of the blood vessel, there's a nucleus of one, and there's a nucleus of another one. They're paving stones. Okay. So this is a drawing which perhaps illustrates that more clearly than the photomicrograph and my scribbling on the photomicrograph. There you see the basement membrane, the subendothelial layer made up of collagen fibers. Don't forget there is that connection between, if there's an injury to the wall of the blood vessel, then collagen fibers get exposed, the blood gets exposed to collagen fibers, and that will initiate the clotting or coagulation cascade. I'm going to go with I think it's the intrinsic pathway. It could be extrinsic, but I'm pretty sure it's the intrinsic pathway, activation by the intrinsic pathway of the coagulation cascade. The capillaries is, as I said, where everything happens. All exchange is happening, osmosis is happening, diffusion, filtration, if there is any, is happening there. Lined with endothelial cells, and then leading towards the venous side of the circulation. But there's quite a bit to capillaries. They have a very important function, obviously. continuously. There are different types of capillaries. There are three major different kinds of capillaries. There are continuous capillaries, fenestrated, and sinusoidal. We'll go through each one or each type. The continuous one form a continuous paved wall. They have tight junctions between the different cells where the cells meet each other, so that it's not easy for molecules to get through in between two cells. This is a continuous type of capillary. The continuous capillaries are typically in and quite abundant in skin and muscle. Just below the skin is an epithelial layer, so it's not actually in the top epithelial cells. It's an epithelial tissue, so as epithelial cells, it's just beneath that you have a lot of capillaries. It's found also in muscles. They form the cells that pave the wall of the blood vessels of the capillaries, form an uninterrupted lining or paving and are held tightly together by these tight junctions. In the central nervous system, there are continuous capillaries, and they have particularly close contact between endothelial cells. So tight junctions are occurring completely around wherever you have an endothelial cell meeting another one, you'll have tight junctions completely in that contact area, so that it's a very tight continuous barrier to molecules resisting the movement of quite a few different types of molecules from the blood into the brain. It allows for diffusion of CO2 and oxygen, and metabolic byproducts can go across, and nutrients can go across, but a lot of the molecules cannot get across into the central nervous system. This, in fact, is called the blood-brain barrier, this tight junction rich continuous type of capillary found in the central nervous system, and can actually be a problem when they're designing drugs to treat the central nervous system, because there are quite a few drugs that cannot cross over through that blood-brain barrier. It's quite a common problem, delivery. Here you see another type of capillary bed. It's a fenestrated capillary bed. This has actual fenestrations coming from the Latin word for windows. So these are actually pores in the wall of the endothelial cell. So this is obviously a much looser kind of, more easily permeable, a more permeable kind of capillary. And this is the kind that you would find in areas of the body that are involved in absorption or filtration mechanisms. For example, the small intestines, you'd find them there. Small intestines are involved in a lot of absorption of materials from the, digested nutrients from the space of the small intestine. You also find them in endocrine glands. Endocrine glands are involved in secretion of product into the bloodstream. And you also find them in kidneys, where you get a lot of tissue that's involved in filtration. You have these specialized capillary beds that are involved in filtering the plasma to form the fluid that will eventually become urine. The third and last type of capillary bed that we'll discuss is they're called sinusoids, and these have very, very large openings within the cell, its endothelial cells themselves, like here and here, for example, or even between the endothelial cells. So you have it's quite a porous type of capillary bed. Large intercellular clefts, large pores within the cell itself, allowing actually the cells of the blood to exit. And this is the kind of thing, very permeable, very leaky. This is the kind of thing that you would find in the capillaries that you find in the liver. where you have the blood actually, interacting with the liver cells directly. They'll directly come into contact with hepatocytes. Also in the bone marrow, you'll have that because blood cells are produced in the bone marrow and have to get into the bloodstream by entering into those capillaries. You see them in lymphoid tissue where you have a lot of interaction between the lymphoid tissue, white cells in the blood, and you find it in some endocrine cells. Allows for very large molecules and even blood cells, and I'm not talking necessarily red blood cells, but any kind of blood cell to pass between the blood and the surrounding tissue. Essentially, the blood will flow into an area of the body that contains these sinusoidal type of capillaries, and the flow will slow down. It'll be quite sluggish. It percolates all through that tissue. As happens with the liver, for example, or the spleen is a good example of that. It occurs also in the spleen. As I said, there are two major different kinds of blood vessels. Blood vessels on the venous side and the blood vessels on the arterial side. And you can easily spot which one's the artery and which one's the vein, not only because they're marked, labeled, but also because of the thickness of the wall, the thick tunica media, lots of smooth muscle here in that area here. You can also, if you look carefully, see the internal elastic lamina, that wavy line. Particularly, I would say up in this area here, I can easily see that wavy line of the internal elastic lamina. And the vein on the left side, which is quite a thin wall, much larger interior diameter, much lower blood pressure on the venous side than on the arterial side. This diagram gives more detail and shows that there's elastic tissue. They've left out the external elastic lamina, but that's okay. So, in fact, veins do contain some muscle tissue, even though it's not apparent. They have a thin layer of tunica media, which can contract in response to certain signals from the nervous system. But in general, it's the arterial side that has most muscle tissue and is mostly capable of much more vasoconstriction and vasodilation because of that. That's an important aspect, not only to directing flow to different areas of your body, but also, as I've said earlier, in maintaining and regulating blood pressure, appropriate levels of blood pressure by adjusting the available space within the overall circulation. And there is some vasoconstriction in the veins, but it's not that significant actually. These are the veins of the body. We probably haven't talked about any of them, although I can say that typically people get blood drawn from their brachial vein. Quite often it's the brachial vein that's chosen to draw blood because it comes down right into the elbow right there, and usually blood's taken from there. Not always, but veins, by the way, are always much more superficial. They're much more closer to the surface than arteries. That's a evolutionary advantage that evolved over time, because if you cut yourself, it's better to have the arteries deeper down, because if an artery gets cut, it spurts and the blood loss is much larger than if a vein gets cut. Okay, now flow of blood back to the heart is called venous return. And venous return actually is what determines how much blood fills up the heart when it's in diastole, and how much blood eventually will become the end diastolic volume of blood inside the heart, right? So that's all determined by is essentially how much blood gets back to the heart. Part of the return of blood in the veins back to the heart is due to compression, due to muscle contraction. So any muscle activity will compress larger veins and cause the blood to increase in its rate of return back to the heart. So venous return goes up. In fact, any of the larger veins throughout the body contain valves to make sure that the blood goes in one direction only, towards the heart. So you can't get any backflow because of those valves. There are many valves all along the larger veins, and they prevent backflow. So you compress a muscle. As soon as you start doing any muscular activity, anything, you're going to start contracting the muscle around veins and increase the return of blood to the heart, the venous return. And here you see a diagram of somebody flexing their biceps, and that squeezes the vein in this vein shown here in their arm, and so the blood is pushed back in the direction towards the heart and cannot move in a retrograde direction because it is stopped by the presence of valves, one-way valves throughout the largest veins. Then when it's a constant pumping action which helps move the blood along through the veins because the pressure in the veins is actually quite low. Overall, the venous side of the circulation is essentially a capacitance part of the circulation. Veins are capacitance vessels. They can hold quite a bit of the volume of the blood. In fact, much more of the overall volume of the blood is within the venous the venous side of the circulation than the arterial side. So 80%, up to 80%. And of course, as I said or pointed out to you, there are one-way valves that help with the venous return. There's a number of things that, in fact, are required to help get the blood back to the heart, and it's helped by three major factors. One is gravity. The other is contraction of skeletal muscles. Whenever they contract, they squeeze the veins and help move the blood along. And also the action of breathing. Inspiration and expiration also help to move the blood along. All right, I think this is a good spot to stop in this particular video. So this is the end of Unit 5A, Part 1.